Hello everyone, this is Minoy Cristobal from STEM Monogastric Nutrition Laboratory at the University of Illinois. Today I'll be discussing the standardized ileal digestibility of amino acids, digestible and metabolizable energy, and standardized total tract digestibility of phosphorus in a new variety of soybeans fed to growing pigs. This work was presented in 2023 ASAS Midwest meeting. Soybean meal is still an excellent source of protein for monogastric diets, and this is mainly due to its high amino acid digestibility. Currently, there is a high demand for soybean oil and soybean meal, and since many years ago, new varieties of soybeans have been developed, and recently, a new variety of soybeans called photoseed was developed. This seed was developed by the Siacal company based out of San Diego, California. For this, the company applied a genetic technology which consists of increased lipid droplet encapsulation, which reduces feedback inhibition of photosynthesis and drive greater carbon capture. As more carbon is stored as lipids, more photosynthate is produced, which promotes the rhizobia activity and increases nitrogen fixation. With this characteristic, there is an increase of up to 18% of oil and up to 3% of crude protein in the photoseed soybeans. However, there is no research about the nutritional value of the photoseed soybean meal that is left after oil extraction for swine nutrition. Therefore, the objective of this study was to test the hypothesis that concentration and digestibility of nutrients are not different in soybean meal produced from photoseed soybeans compared with conventional soybeans when fed to growing pigs. The ingredients used for this study were the conventional soybeans mechanically expeller processed and the photoseed soybeans mechanically expeller processed. Now let's take a look at the nutritional composition of the ingredients. Color green will be used to represent the values for conventional soybean expellers and orange the values for photoseed soybean expellers. We observed no large differences between the two ingredients, and for gross energy, the values were around 4,800 kcal per kilogram. For crude protein, the values were around 44%, and for crude fat, around 7%, and for total dietary fiber, the values were around 23%. Not much difference for total calcium, and slightly greater numerically values for total phosphorus and non-phytate phosphorus. For total amino acid concentration, slightly greater values in the photoseed ingredient compared with the conventional ingredient. Slightly lower values for sucrose, stachyose, and raffinose in the photoseed ingredient, but also this ingredient contained more trypsin inhibitors compared with the conventional ingredient. Now that we have seen the nutritional composition of the ingredients, let's move on to experiment 1, where the objective was to determine the standardized ileal digestibility of crude protein and amino acids in conventional soybean expellers and photoseed soybean expellers fed to growing pigs. For this trial, we used 3 diets, a nitrogen-free diet and a diet containing conventional soybean expellers or photoseed soybean expellers as the only source of amino acids. We used 9 cannulated barrows of around 11 kilos that were fed 3 times metabolizable energy for maintenance, and the animals were allotted in a 3x3 Latin square design for a total of 9 replicated pigs per diet. Each period consisted of 5 days of adaptation and 2 days of ileal sample collection. For the statistical analysis, we used the mixed procedures in SAS with LS means statement and pdiff option. The diet was the fixed effect, and the square, period, and replicate were random effects. We considered 1 pig as our experimental unit. Let's take a look at the results for experiment 1. We're looking at the standardized ileal digestibility of crude protein and indispensable amino acids. The values were greater for crude protein, arginine, isoleucine, and lysine in the conventional soybean expellers compared with the photoseed soybean expellers, and no differences for the other amino acids. Then we used the coefficients obtained and multiplied those by the total amino acid concentration in ingredients and obtained the concentration of digestible amino acids. In this case, no differences were observed between the 2 ingredients, but for arginine, lysine, methionine, and tryptophan, greater values were obtained in the photoseed soybean expellers compared with the conventional soybean expellers. For experiment 2, the objective was to determine digestible energy and metabolizable energy concentrations in conventional soybean expellers and photoseed soybean expellers fed to growing pigs. For this trial, we used 3 diets, a corn-basal diet, a diet containing corn and the conventional soybean expellers, and a diet containing corn and the photoseed soybean expellers. We used 30 barrows and yields of around 18 kilos. The pigs were allotted in a complete randomized design for a total of 10 replicated pigs per diet. We used the marker-to-marker approach, and each period consisted of 5 days of adaptation to the diet and 4 days of urine and fecal sample collection. The statistical analysis was similar to the previous experiment, and the diet was the fixed effect and replicated pig was the random effect. Now let's take a look at the results for experiment 2. We're showing the apparent total tract digestibility of gross energy of diets. We observed no differences among the conventional soybean expellers, the photoseed soybean expellers, and corn. And the total tract digestibility was around 90%. Now looking at the digestible energy and metabolizable energy concentration in ingredients, we observed that the values were greater in the soybean expellers compared with corn for both digestible energy and metabolizable energy concentrations. This difference was mainly due to the greater fat content in the soybean expellers. And when comparing the conventional and the photoseed soybean expellers, no difference were found for digestible energy and metabolizable energy concentrations. Now let's move on to experiment 3, where the objective was to determine the standardized total tract digestibility of phosphorus in conventional soybean expellers and photoseed soybean expellers with 3 phytase levels fed to growing pigs. For this trial, we used 6 diets in a 3x2 factorial arrangement. One factor had 3 levels, which were the 3 phytase levels, and the other factor had 2 levels, which was the type of ingredient, either conventional or photoseed. We used a total of 48 barrows and gills of around 12 kilos. The pigs were divided in 2 blocks of 8 replicated pigs per diet, and we used the marker-to-marker approach, and each period consisted of 5 days of adaptation and 4 days of fecal sample collection. The statistical analysis was also similar to the previous experiments, where the diet was the fixed effect and the block and replicate were the random effects. Let's take a look at the results for this experiment. We're looking at the standardized total tract digestibility of phosphorus of ingredients. These values were obtained after correcting the apparent total tract digestibility of phosphorus for the basal endogenous losses of phosphorus as suggested by NRC. There was no significant interaction of source and phytase, and there was no significant main effect of source, but there was a significant main effect of phytase, and we can observe that in the graph by looking at the standardized total tract digestibility values and how that increased as we increased the phytase levels in the diets. Based on the results of the 3 experiments, we conclude that photoseed soybean expellers compared with conventional soybean expellers had similar nutrient concentrations, lower digestibility of some amino acids, but no difference in digestible amino acid concentration, had no different concentration of digestible energy and metabolizable energy, and had no different digestibility of phosphorus with or without added phytase. And the results also imply that for oil production, it might be more economical to produce photoseed soybeans due to the higher oil yield, and the photoseed soybean meal obtained after extraction could be used as efficiently as conventional soybean meal and obtain similar responses on pig performance. With that, I would like to acknowledge the COCAL company and Dr. Hans Stein Monogastric Nutrition Laboratory. Thank you.